while people in the room are thinking about their questions, I've got lots coming through online. So it's great to have so many clinicians on a panel. We have a lot of bioinformaticians in the audience here at London Calling this year. And one of the questions that we have coming through is about the role of the bioinformatician in many of these extraordinary clinical initiatives. So could I perhaps ask a couple of you to comment on the role of the bioinformatician? Is it around creating more automated analyses and pipelines for clinicians to use, or do you work directly with bioinformaticians as part of the team? Perhaps could I invite Jessamine, do you want to go first?
Sure. So prior to 10 years ago, I had no idea what a bioinformatician does, and now I'm completely addicted to having a big team of them. So I think obviously the automation is incredibly helpful and important, and in terms of speed, especially when you're used to having something that you can then automate and get it quicker. But when you're talking about discovery, I feel like I am not, me and my clinician colleagues, we cannot make use of the data without having a bioinformatician speak to us. So our meetings are, all of the data is presented by a bioinformatician and explained to us in a way that makes sense clinically, and then we have a big discussion back and forth. So actually that exchange of information and expertise with our bioinformaticians has been incredibly important and helpful to make the most out of this data for patients.
Thank you. I'm going to just allow the audience, perhaps, if you could put your hand up and then stand up, perhaps, to ask the question. If you let people know where you're from as well.
Okay. Hi, sorry. Hi, Luca Mazzella. I'm an oncologist, actually, from Italy, and I'm going to ask, actually, the same question that I asked for the same speaker, the first speaker of the conference, to our oncologist colleague. So the issue with nanopore sequencing is that the sampling procedure has to be modified from the beginning, because as far as I know, the sequencing is not very compatible with what is routine and clinical diagnostics, which is FFPE. So I'm curious to understand what you guys, I mean, have you implemented a dedicated procedure for the sampling, based on the presence of this particular bottleneck? Do you see that this can really be implemented on a wider basis, or only in centers that have the possibility to modify the sampling procedure according to the type of analysis that they have to do downstream? Thanks.
Thank you.
So if I could just summarize to make sure I understand, your question is about the fact that we need fresh tissue to do this properly, as opposed to how most tissue is collected, which is paraffin embedded. And that is true, that's a struggle that we've had. However, it's usually a clinical issue. When I ask patients whether they would be willing to have a biopsy so that I could better understand their cancer and we could better potentially identify treatments for them, almost no one has ever said no to that. So it is a problem to get fresh tissue. It requires extra procedures, there's extra risk, it can add time. So I don't think that we can, I'd love to be able to change the way that all tissue is sampled. I don't think that's realistic. So we do focus on a fresh tissue biopsy, and I find that it's really clinicians that have the problem with that, and then the logistics of organizing it. That being said, we have been looking at short-read sequencing for FFPE embedded materials, and we are working on ways to minimize the errors in that data set. I'm sort of optimistic that we could identify a cohort of patients that we could use FFPE on, so perhaps that would be translatable to this technology as well. That would be beyond what I know. But Thanks. For now, yes, fresh frozen tissue.
Thanks, Janessa. I'm going to ask a different question now, and perhaps invite Dr. Ahmed to comment on this, which is, You've presented some findings from your work in the Middle East, and there's a question online about how applicable you think these findings are from specific ethnicities to wider populations. And could you perhaps comment on whether it's crucial to sequence all cultures and ethnicities to obtain disease-relevant and clinically important information?
Well, that's a lot of information. Yeah, I mean, definitely I think other colleagues have presented their work from other populations showing the value of long-read sequencing and adding more diagnoses beyond short-read sequencing. So I definitely think it's beyond, I think it's going to apply to any population with rare diseases. I think the second question about sequencing, definitely, I think we need control data sets. That's which also hinges on the question about the bioinformatics. I think they are the main players with this because we generate a lot of new data. For example, 20, 25,000 structural variants. There are multiple, callers, variant callers, and I think someone presented on how each one of those is performing. So this is all work in progress, and we rely a lot on bioinformaticians and optimizing these callers. But also, once we get the variants that we think might be real, we need to filter across a population-specific data set, right? So in sequence variants, we use Nomad, which has 120,000 or 140,000 individuals, which unfortunately lacks Middle Easterns. But we need population-specific data sets for SVs, CNVs, and even SNVs in our setting to be able to clinically plug this in as soon as we find variants, to filter out common variants in the population and just focus on the ones that are going to be unique to the patient. So that's a major challenge. Yeah.
Thank you. Now, I have a question that I'm going to invite Emma Bapel, as a fellow clinician here in the NHS, to comment on, which is that some of your use cases were extremely powerful in the talk that you presented, so thank you for sharing those. I'd be really curious if you could comment on what you think it would take for wider adoption to happen and the genomic revolution to really have an impact across the NHS, to build on some of those powerful use cases you presented.
In what sense, Emma?
For it to be more widely adopted into mainstream clinical care in the
Oh, you mean genomics to be more mainstreamed?
Yeah.
I mean, I think we've made massive progress in that area in this country. We've got a nationalised genomic medicine service. And in all honesty, the majority of the requests that come in now are coming in to us from paediatricians. All of the requests that come in for the rapid testing service that is done in Exeter need to be approved by clinical genetics. But the reality is the impetus to make those requests now entirely comes from the paediatricians that are in intensive care or on the neonatal units, because it's them that's dealing with those patients, and it's them that will have to see the impact of those results directly. So I don't think as a clinician anything becomes real to you until you, or you don't see the importance of it until you see the value to your patient. Mm-hmm. So the more and more of these sorts of powerful stories we have, you can't ignore that as a doctor. You want to do the best that you can for your patient. So showing the results of this sort of work is what makes people want to engage with it.
Thank you, Emma. So we have time for one more question in the room. I can invite you to put your hands up and we'll get a microphone over to you. I have one in the middle over here.
Thank you to the panel. I'm John from Stanford. As Dr. Ahmad was alluding to in his previous comment, we rely heavily on clinical genetic databases in order to make final diagnoses. And with the genetic diagnostic rate that we see with whole genome sequencing, we're somewhere around 40% when we think about our infants. As these genetic databases continue to grow, especially with structural variation or DNA modification, things that Nanopore is really good at identifying, do you have a strategy in order to reanalyze genomes with negative findings? And if so, can you speak to that?
I don't think we're thinking that far yet. We are still, doing a pilot study on 50 genomes. And I think we're still sifting through data to get the best out of it. And like I said, we're struggling in filtering the most relevant information. As you know, we don't have any baseline data set to compare to. And I definitely think as soon as more databases become available, will be much easier to filter through variants. And I also think as these pipelines are maturing, will be helpful to go back to the raw data and reanalyze negative cases. But I honestly we haven't thought that far ahead. And even for our clinical testing with short-read sequencing, we haven't done any systematic negative exome reanalysis. It's just usually you have to have, as a clinical lab, especially for us, we're a new small lab that's growing, you need a specific infrastructure to be able to do this systematically for all your 1,000+ negative cases and report back to clinicians and physicians. So I think hopefully we'll get to that point. But it's a great question.
Please join me in one last thanking of our panel for your contributions this afternoon.